What if I told you cutting-edge technology could potentially provide a way to cure diabetes, prevent blindness, and eradicate HIV, saving countless lives? These are merely 3 of the numerous illnesses that could be thwarted away by the use of the genetic editing technology CRISPR-Cas9. Gene editing technology, known as CRISPR-Cas9, is actually a complex made up of the sequence CRISPR and the associated protein Cas9. According to the Broad Institute, CRISPR, clustered, regularly interspaced, short palindromic repeats, is named for its characteristic repeating patterns and forms the base for CRISPR-Cas9. Cas9 is an associated protein, meaning it interacts with the CRISPR sequence to complete the edit. Although this complex relies on both pieces to alter DNA, CRISPR and Cas9 were discovered at different times. In 2010, according to the National Institute of Health, through thorough research and experimentation, it was discovered that this genome editing technology alters DNA sequencing by cutting the DNA strand. First, CRISPR is translated into short strands of RNA, the messenger for creating more DNA, and looks for DNA as it would in ordinary transcription and translation. However, once the CRISPR RNA finds the DNA, it releases the Cas9 enzyme, which then binds to the DNA and cuts the sequence at the precise location. So what does all of this have to do with you? Well, in 2013, the CRISPR-Cas9 system began testing on human and animal cells. Testing on animal and human cells began in 2013 at MIT in the Zhang lab. This was the first lab to publish findings on the processes of editing mice and furthermore human cells. According to the National Cancer Institute, experimentation has begun on mice cells to develop more effective treatments and potential cures for cancer with the use of CRISPR-Cas9. Headed at the Memorial Sloan Kettering Cancer Center in New York, experiments have been run using CRISPR to modify immune cells to kill cancer cells more efficiently. The results of the experiment showed that immune cells modified by CRISPR killed more cancer cells than immune cells treated with conventional methods. With the Harvard School of Public Medicine, the Memorial Sloan Kettering Cancer Center are developing potential ways in which CRISPR can make cancer treatments more effective with tumor reduction. Although these methods are not yet available for patients, they are quickly moving towards clinical trials according to the National Cancer Institute.
So this allows for that really specific DNA repair or change. So if you have a certain disease that is encoded in certain nucleotides, you could actually target those individual nucleotides to be changed. And for reasons for genetic exploration, like what I do, we could actually modify the genome of the organism that we're working with to express different proteins, more of them, less of them, see what things are interacting, that sort of thing.
Modified animals could potentially pose safety threats to both humans and the environment. Even the director of U.S. National Intelligence, James Clapper, weighed in on the matter at the annual Threat Assessment Report in February of 2016, saying that the easy access, low cost, and speedy development of genome editing could increase the risk that someone will engineer harmful biological agents. Technology like this thus poses potential for complications, as the effects of using CRISPR-Cas9 are unknown and unpredicted. Despite the negative possibilities, the reason scientists have even pursued the CRISPR-Cas9 system is because of the overwhelming benefits with regards to animal and human engineering.
This is really important. I think it's going to be very important for for health. I think there's going to be a lot of personalized medicine that comes out of this. Basically, you and I have different genetic codes, and based on that, we are more likely or less likely to get certain diseases. So if we knew that ahead of time, you could actually personalize medicine to change those parts of your genome to have less of an effect later in life.
For example, many people can't receive certain vaccinations because they're produced commonly with chicken eggs. Biologists from Commonwealth Scientific and Industrial Research Organisation in Geelong, Australia, have said they can fix the allergic reaction by genetically modifying chickens to lay eggs without the bacteria. This would economically and effectively solve a common vaccination issue and give people the chance at a healthier life. While we have seen the works of CRISPR primarily in animals, some scientists have begun to apply this technology to embryonic human cells as well.
One of the major concerns, other than the ethical implications of this, which have yet to be explored, there are some technical ones, such as off-target effects. So, because CRISPR is so specific, is very specific, it uses 20 nucleotides of surrounding sequence to target a site. However, in the human genome, if you have repetitive sequences, that means that Cas9 might be cutting in different parts of the genome, and the field is doing its best to eliminate off-target effects as much as possible, because when you don't want that to happen, but that's going to be a big hurdle going forward with any sort of testing.
According to a review done by MIT Technology, a team of Chinese scientists used CRISPR in 2015 to correct a gene defect that resulted in a fatal blood disease by editing the DNA of the fertilized eggs. Although the experiment itself was promising, the results were considered unsuccessful, given that CRISPR seemed to work for some cells and in others produced no results and even unexpected mutation. Previous to the experiment, people were concerned about germline modification, referring to the potentially harmful mutations future generations may face if a DNA is altered to express a specific trait.
What certainly raises ethical implications is whether that sort of change should be made in eggs or sperm or embryos, where those changes could then be passed on to the next generation. So I feel that it's appropriate with guidelines to conduct research in those kinds of systems, even in human embryos. I don't think that it would be appropriate to use this technology, at least today, for any kind of clinical application in human embryos. The science is going 1,000 miles an hour. One of the roles that we as scientists need to play is to really communicate the power of this technology and how we can be responsible in using it. For that conversation to carry weight globally, it really needs to be a conversation that involves scientists that are international. Do we know enough about the human genome to understand the impact of making changes to it in a developing embryo? And so I think that there I would like to see our society draw a line and say that, we won't go there right now. We'll take the time that we need to really think about this.
Introducing CRISPR technology to human embryos begs the idea of designer babies. The term designer babies has been used to describe what many parents would use the technology for, determining the skin tone, gender, and physicality of their child. Not only does CRISPR present an ethical dilemma of engineering humans, but there's also reason to believe more social injustice can evolve. What groups of people are going to be able to design their babies? It's likely, considering the cost of this technology, only the wealthy will be able to do so, leaving lower-class families behind in an evolving human population. From this, humans could be more intelligent, efficient, and advanced, leading into a whole new society with redefined class structures and social statuses. Could it be that these individuals would stand at the forefront of society? Although the opportunity to prevent and eliminate disease could change lives for the better, where do we draw the line for genetic engineering? Ethically, should humans be allowed to play God? And scientifically, should scientists even chance this technology if there's always a threat of negative mutations that could be irreversible? CRISPR-Cas9 can be seen as a modern-day Pandora's box, as it contains potential unknown consequences, but still leaves hope for a stronger and healthier humankind.